I want to thank everyone for joining us today. My name is Andrew Buis. I work as a senior formulation scientist at Altasciences. Today, we're going to be talking about the advantages of terminal sterilization over aseptic manufacturing. As a co-speaker, I'm here with Derek Prince.
Thank you, Andrew. I am Derek Prince, and I'm the president of Prince Sterilization Services.
So the learning objectives for this webinar, we're going to give a basic background of aseptic manufacturing and its importance, the benefits of terminal sterilization, the regulatory expectations behind terminal sterilization, valuation, and a case study. My name is Andrew Buis. I've been working as a formulation scientist for several years. I've been with Altasciences for about 6-7 months now, and previously I worked at Lubrizol Life Sciences.
And I'm Derek Prince. I'm the president of Prince Sterilization Services. I have extensive experience in steam, dry heat, and low temperature sterilization, ready-to-use pharmaceutical componentry, as well as laboratory testing. I have a master's degree in molecular virology and a PhD in microbiology.
So Altasciences at a glance. We advance drug development from lead candidate selection to clinical proof of concept and beyond. As a mid-sized CRO, we have over 25 years of experience in early drug development. We have strong leaderships and scientists from pretty much all industries, including pharma, biotech, academia, and CROs. We use state-of-the-art facilities to provide the capacity and harmonized workflows to support your clients' needs, preclinical, clinical pharmacology, and bioanalytical needs. Whether you have a single study or a complete program, our goal is to simplify the process to speed and ease. We at Altasciences, we pick up a client's needs from the preclinical stage, going all the way to phase 4 and marketing. So we at Altasciences stretch from coast to coast, covering preclinical, clinical, manufacturing, and bioanalytical and research facilities. So this is where Altasciences comes in. Our capabilities offer a purpose-built FDA-inspected GMP facility, including multiple manufacturing grade B suites, cutting-edge grade C suites, including segregated and dedicated potent handling areas. This is our site located in Harleysville, Pennsylvania. Our suites can handle research and formulation development to commercialization with multiple production capabilities. Our team offers drug development, manufacturing, and analytical services, including formulation development, phase 1 through commercial manufacturing and ICH stability, storage, and testing. We also provide analytical method development qualification and validation, as well as finished product and release testing. We have formulated, tested, and manufactured nearly every currently available pharmaceutical dosage form on the market, including lipid and powder-filled capsules, over-encapsulated capsules, nanomilled suspensions, solutions, creams, gels, powders, and terminally sterilized injectables in our grade C and D clean rooms.
Thank you, Andrew. Prince Sterilization Services is extremely proud of our long history servicing the pharmaceutical and related life science industries. Our company was founded by Dr. Herbert and Leah Prince in 1970 as a contract testing laboratory. It was taken over in the early 90s by Dr. Daniel Prince, who then spearheaded the company's expansion and diversification efforts, ultimately positioning Gibraltar as an industry leader for quality contract laboratory testing. In 2015, we added a sterilization services division, and in addition to laboratory testing, began offering contract sterilization services to the pharmaceutical markets. In 2018, we sold off Gibraltar Laboratories to Nelson Labs and spun off the sterilization services division into its own entity. Today, Prince Sterilization Services is independently owned and operated by the Prince family and has recently completed a 30,000 square foot expansion to better service our pharmaceutical customers. At Prince, we kind of divide our business into two main functions. The first one I'll talk about on the right is the ready-to-use pharmaceutical componentry side. So we sell to CMOs, similar to Altasciences, vials that are ready to use and ready to fill with their drug products, whether it's in a tray or in a different type of format. We're able to supply those products in a ready to use state, as well as the stoppers and seals as well. So a drug product can just be filled inside without the need for the washing and sterilization and deprogenation equipment to achieve a sterile container. More in line with this presentation today, the other arm of our business is that of contract sterilization. The contract sterilization side, which I'll primarily talk about as it relates to this presentation, is the use of steam sterilization to terminally sterilize drug products in filled containers, whether that's a blow fill seal type container, a glass or plastic vial, a syringe, or an IV bag.
Thank you, Derek. I will now go over aseptic manufacturing and our capabilities at Altasciences. There is a growing demand for sterilization of small molecule products, including heart medications, eye drops, analgesics, and antibiotics, as well as common intravenous solutions such as glucose, potassium, and saline. Many products are used by medical professionals and hospitals to ensure patient safety. The drug products and related instruments and materials are sterilized. This ensures that there are no microbial contaminants like fungi or bacteria present in the products used. With that in mind, there are several modalities for sterilization. We have aseptic manufacturing versus terminal sterilization. The reasons to use aseptic manufacturing. Aseptic manufacturing is important to produce a sterile product. This is primarily used when terminal sterilization is not possible. And the reason as to why this is a secondary option over terminal sterilization is the fact that aseptic manufacturing is very costly, with very strict validation requirements needed to achieve SAL, and cannot provide the same quantitative levels of sterility insurance as terminal sterilization. So it should be used after all other sterilization options have been exhausted. However, this is unavoidable in many cases. Methods of sterilization are highly dependent on the active pharmaceutical ingredient or API used in the drug. And aseptic manufacturing is only used when those conditions cannot be met. So aseptic manufacturing advantages. Sterility is controlled on every aspect of manufacturing. Each component, drug container, closure, primary, secondary packaging, in some cases, every excipient and API is individually sterilized first, then carefully assembled in a dedicated clean room with a highly controlled environment to make the finished drug product in a manner that prevents contamination. Different types of packaging components also undergo sterilization cycles. However, not all packaging can be controlled this way. So aseptic manufacturing controls. There are 4 key aspects that controls aseptic manufacturing. First, facility design and HVAC validation. The second, environmental monitoring. The third is personnel training and monitoring. And the fourth, but not least, is process simulation, also known as media fills. Although they cannot provide the same level of SAL as terminal sterilization, several aspects of control can minimize the risk of contamination. Dedicated clean rooms are needed and designed with a strictly controlled environment to make the finished drug product in a manner that prevents contamination. There is an inverse relationship to the number of particles allowed to the amount of engineering controls and validations needed. In essence, the number of free-floating particles allowed in a clean room, the lower the amount of particles, the more engineering controls and validations are needed to qualify said clean room. In addition to traditional clean rooms, barriers can be used to separate the operator from the fill area. These can be full isolators that are completely closed to the surrounding environment, or RABs, remote access barrier systems. These both full isolators and RABs require operators to interact with the fill area only through special glove ports, further minimizing the potential for contamination of the drug product. In all cases, operators are required to have full coverage, no hair or skin exposed, with sterile coveralls, hoods, boots, goggles, and gloves. and must be highly trained in all relative techniques. Each operator must pass certification testing before they can participate in aseptic manufacturing. The entire aseptic process is routinely simulated in a challenge that is designed to detect the potential for contamination in the finished drug product. A single unit of positive growth is a sign that the process has failed and needs to be thoroughly investigated. Zero positives is the norm for this challenge. So aseptic manufacturing is based heavily on the clean room. Clean rooms are engineered spaces to control airborne particulates. Clean rooms come in many grades, but they're usually based on the number of particles allowed. HEPA filters are used to control the airborne particulates through a laminar airflow system. They are also designed to keep outside influence minimized through positive air pressure and airlocks. Clean rooms are well isolated, well controlled from contamination, and actively clean. They are designed to keep everything from dust to airborne organisms or vaporized particles away from whatever product is being handled or manufactured inside of it. These rooms are graded based on the number of particles allowed at rest and in operation. They maintain their particulate-controlled air using HEPA filters. HEPA filters are effective at removing up to 99.97% of particles greater than or equal to 0.3 microns. However, more effective HEPA filters are required for higher grade clean rooms. Higher air pressure is used to create and maintain a positive pressure in the clean room to prevent outside air from coming in. A series of airlocks are also installed to reduce outside influence as operators don gowning and enter the clean rooms. The picture on the right is a basic concept of the work inside of a clean room, of what work inside a clean room looks like, and also the airflow. We at Altasciences manufacture most of our products in our grade D suite. We do have a grade C suite, which is specifically designed to handle highly potent compounds and products sensitive to contamination on a large scale. What qualifies our clean room as a grade C or ISO 7 for international standards is the maximum number of permitted particles above 0.5 micron and 5.0 micron. In accordance with US and international standards, our grade C clean room has no more than 352,000 particles between 0.5 microns and 5.0 microns at rest and 3.52 million particles in operation, as well as no more than 2,900 particles between 0.5 micron and 5.0 microns at rest and 29,000 particles greater than 5.0 microns in operation. This reduces exposure to potent compounds during manufacturing. So to continue into how we manufacture in our grade C suite, multiple products, both potent and non-potent, are manufactured in our grade C suite with the intent of limiting microbial contamination and operator exposure. We also utilize sterile filtration and terminal sterilization as a way to ensure sterility. In terminal sterilization, the final drug product is passed through a filter that has itself been terminally sterilized and designed with appropriate pore sizes, surface chemistries to remove bacteria via size exclusion, entrapment, electrostatic interaction, and other modalities. The filtration step of the formulation drug product manufacturing area must be validated by demonstrating repeatedly that passing the product through the selected filters removes microorganisms below a defined level. For this validation, a bacterium of very small size, even compared to other bacteria, which is therefore more likely to pass through the filter, is used. Self-filtration is mostly applied to solutions, some suspensions based on particle size, solvents, and gases. It is necessary to limit the contamination and bioburden prior to terminal sterilization. And again, we use these to physically remove the microbes by passing the material through a membrane. Once the products have been sterile filtered, we often package the products and ship them out for terminal sterilization. So terminal sterilization from a CMO/CDMO perspective. As a CMO/CDMO company, We manufacture a wide variety of products using an assortment of distinct APIs for virtually every dosage form based on our clients' needs. In many cases, the finished products will need to be terminally sterilized when sterility for manufacturing cannot be guaranteed. As such, we often employ services that provide different forms of terminal sterilization. For products that are thermally sensitive, we contract out companies that terminally sterilize through radiation, i.e., gamma rays or electron beam, as it is effective for sterilization through microbial DNA destruction. For products that are thermally stable, it is highly recommended that we use steam sterilization, as it tends to be the most effective approach to achieving product sterility. This is where Prince Sterilization Services comes in. Prince Sterilization Services have a long, prestigious history of gas/heat-based terminal sterilization. And with that, I will pass off to Derek to explain terminal sterilization.
So now to talk a little bit more about terminal sterilization and how it relates and where it fits in the process related to aseptic fill or sterile filtration aseptic manufacturing. As defined by the PDA Technical Report number 1, terminal sterilization is a process whereby product is sterilized within its sterile barrier system, the only way to provide a SAL or a sterility assurance level to a finished drug product. Some key words that I think are important for everybody to kind of know or be aware of as you listen to this presentation are on this slide. Just real quickly, I'll talk about items being sterile, which means free from viable microorganisms. Sterility is the state of being free from viable microorganisms. Sterilization refers to a validated process used to render product free from microorganisms. I'll talk about bioburden. Bioburden is the population of viable microorganisms that are on or in a product or within a sterile barrier system. You'll hear sterility assurance level, synonymous term SAL with a PNSU, a probability of a non-sterile unit. That implies the probability of a single organism occurring on an item after it's exposed to a sterilization process. And then I'll talk about F sub 0, which I have another slide for this as well, which is a term used to calculate the equivalent lethality at a temperature other than 121 degrees Celsius with a Z value of 10. Not here is something called the D value, but I'll talk about that more as well. I understand that we may have a technical audience, which is good, but I also understand that some people may not be overly technical with the concepts that will be discussed today. So I'll do my best to kind of bridge the gap between making sure I'm addressing technical audience that's here in the room, as well as some people who are a little bit less experienced with some of the topics that I'll be discussing. Essentially, what I'll be discussing is steam sterilization, moist heat steam sterilization, autoclaving, steam sterilizers. Those are kind of all synonymous terms to the steam sterilization topic that I'm talking about. So you may hear me refer to them interchangeably. And typically in an autoclave or a steam sterilization type system, the process is overly simplified for everyone's information on this slide, but essentially how it works is you have an autoclave or a steam sterilized chamber, you're removing the air, you're injecting steam, and then that steam is injected, within a predefined temperature, pressure, and time relationship. That causes the sterilization of the load items, which I'll show this more in more detail later on. And after that contact time is there with the steam's removed, the load items are dry. And that's essentially how steam sterilization, that's essentially what I'll be talking about today as it relates to steam sterilization. There are common steam sterilizers or autoclaves on the market. I'm going to show a few of those on these slides here just to kind of get everybody familiar with what we're talking about. The one on the left is a small benchtop unit. If you've ever been to a dental office and you peeked in some of their back rooms, you may see one of these. They use these to sterilize the hemostats and scissors or forceps that they use in their offices. On the right is a more standard, what would be called a standard hospital unit. So if you go to a clinic, a medical clinic or a hospital, they'll have these sterilizers that look like large refrigerators, and they'll fit trays and other surgical tools or other ancillary components used in hospitals that need to be sterile. At Prince, we also have several large commercial GMP steam sterilizers or autoclaves, and these are large sterilizers used to support more commercial type accounts and commercial projects, similar to what we'll be talking about today, finished drug products. So this is some of our equipment, and the unit I'll particularly be talking about today is this sterilizer in front of us. And this is a specialized sterilizer that allows us to sterilize liquids in sealed containers. So with respect to terminal sterilization, it's really broken into two different forms. And the USP does a really good job of differentiating what those two different forms of terminal sterilization are. The first one, which is defined in the USP chapter 1229.1, is direct contact. And in this example, just as I described before, again, air is removed and steam is injected. Now, that steam that's injected is going to directly contact the load item that you want to be sterile. So with respect to terminal sterilization, we now know that that means that the load item is contained within a sterile barrier system. So that steam needs to penetrate the terminal sterile barrier system in order to make a direct contact with that load item. So on the top right of the screen, if we see two examples, we have stoppers and seals. So these stoppers are within a sterile barrier system. It's a Tyvek HDPE bag, and the steam is able to pass through the Tyvek, contact the load item. On contact, the steam condenses, and it's that condensation, that condensing, the transfer of energy at that step is what causes the lethal effect of steam to the microorganism that could be contaminating the product. And then that steam is able to flash back off and go back through the packaging and essentially dry the load. So in this example, we have steam directly going through the package, contacting the load item, and then being steamed off, flashed off, and dried. And that would be terminal sterilization as direct contact. And what you're left with is the stoppers or the seals in its final sterile barrier system, ready to use. Now, the other side of terminal sterilization, as defined by the USP in USP 1229.2, is moist heat sterilization of aqueous liquids. So of course, as we know that terminal sterilization means sterilizing something in its final sterile barrier system, how do you accomplish that if that sterile barrier system is not permeable to the sterilization agent? So of course, this uses a different approach as liquids in sealed vessels, whether it be a vial or a syringe, an IV type bag, those types of commonly used vessels that drug products are filled into. How do you sterilize that? Of course, we know that the steam that you supply to the system can't penetrate a glass container, so that's not going to cause the sterilizing effect. So how this process works is, if we use this vial as an example, the steam applies heat to the outside of the container, and the heating of that container wall results in the heating of the internal volume. And that provides a heating effect that would sterilize and kill any contaminating organisms within the liquid. Similarly, like direct contact, due to the temperatures that this container is going to be exposed to, and due to the pressure controls and things like that, that would be controlled by the user, there will be a level of boiling of the aqueous product, because remember, this is an aqueous solution. There will be a level of boiling going on internally, which will supply steam that may contact any contaminating organisms as well on the inside of the container. Terminal sterilization that we're really going to focus on in this presentation is the terminal sterilization of liquids in sealed vessels, and not so much terminal sterilization of stoppers and seals and any type of medical device that would be in a sterile barrier system. To sterilize a liquid in a sealed vessel, using the vial as an example, and to be able to expose that to a terminal sterilization process, it requires a specialized autoclave or a specialized sterilizer, which we have at Prince. And essentially, the reason you need a specialized sterilizer is because as you heat a sealed liquid, the heat is going to cause the liquid to expand. And as that liquid expands inside the container, it results in a higher pressure within that container than the external environment. So we have a specialized chamber at Prince that allows us to apply an air overpressure to the outside environment. And what that does is it allows the container to equilibrate and prevents the container from either, one, prevents the glass or the plastic container from exploding, or two, prevents the seal integrity system from failing, both of which would cause big issues. Now that we have a little bit of a better understanding about what terminal sterilization is and the type of terminal sterilization we'll be talking about in this talk, what are the regulatory expectations? What do the regulatory authorities expect with respect to terminal sterilization of sterile filtered or aseptically prepared drug products? I have a few examples here to share with the room because it can be a little confusing or challenging navigating all the different documents that talk about these types of activities. So the first one I'll just read is from Annex 1, most recent revision and update for the manufacture of sterile products. And here they claim, where possible, finished products should be terminally sterilized using a validated and controlled sterilization process, as this process provides a greater assurance of sterility than a validated and controlled sterile filtration process and/or aseptic processing. Where possible, heat sterilization is the method of choice. From the FDA, guidance of industry, it is a well-accepted principle that sterile drug products should be manufactured using aseptic processing only when terminal sterilization is not feasible. A few more here from USP 1211, terminally sterilized products are the lowest risk category of sterile pharmaceutical products. Unlike products aseptically manufactured under conditions designed to prevent microbial ingress, terminally sterilized products are subjected to a sterilization process that imparts a quantifiable safety level. From the European Agency, evaluation of medicinal products, heat labelability of the packaging material should not in itself be used to disqualify a product from heat sterilization if it is otherwise heat stable. And then another one from Europe, if a decision is made to not terminally sterilize the drug product, drug manufacturers should be prepared to provide scientific explanation and justification for making such decision. It's clear how regulatory authorities view the usage of terminal sterilization and what stakeholders should keep in mind when evaluating terminal sterilization or when evaluating their aseptic or sterile filtration of their drug product. So with that thinking, you would think that terminal sterilization is quite commonly used frequently for sterile filtered drug products. But however, that's not the case. In a nice paper by Agaloko and Akers, which I've cited here, they estimated that about 85% of sterile pharmaceuticals are made using aseptic processing. So why isn't terminal sterilization used more, despite the strong language and despite there being no disagreement that applying a terminal sterilization step after the aseptic filling process provides benefits to the patient and more safety? And it seems that there's the use of terminal sterilization is being artificially constrained by unreasonable expectations that the industry is using with respect to time and temperature of the sterilization process, speaking for steam. So there's this fixation on the idea that the destruction of a high population of organisms using a very resistant biological indicator to the process is required to appease the regulatory authorities and to claim terminal sterilization. They state in this paper that the origin of this is unclear, but it's essentially resulted in sterilization conditions that are extremely harsh on drug products, and that being time and temperature relationships of 15 minutes or more at 121 degrees Celsius. So of course, with that thinking that you need to have this, which is called overkill in the industry, you need to have this overkill type mentality to qualify a product for terminal sterilization. That thinking appears to have limited the use of terminal sterilization to only extremely heat stable formulations. So the theme throughout this presentation is kind of under the guise of, can we do better? Is there a better way to do that? And is this really the right way to be thinking about a terminal sterilization program with an overkill type thinking? So to kind of understand that a little better, I want to spend just a quick minute on the decimal decay time, or the D value. So this is another important definition for everybody to understand. And essentially, this is defined as a resistance. So this is the resistance of a particular organism at a particular temperature. And further, it's the amount of time needed to reduce an organism 1 million CFUs or 1 million colony forming units here. Now, if we had an organism that had 1 million colony forming units and it had a D value of 1 minute at, we will say, 121 degrees Celsius is the temperature we're talking about, because we're talking about steam. So if we had an organism with 1 million CFUs and the D value was 1 minute, then after we exposed these 1 million organisms to 1 minute of steam at 121 degrees Celsius, we would expect a 1 log reduction. So we would go from 1 million to 100,000. And then that process continues. So then as you go from 100,000, as you apply another minute at 100,000, and you have 2 minutes total, you'd go from 1 million to 100,000 to 10,000 in 2 minutes, and so on. So that's what the D value means. The other term that's closely tied and closely used, and I'll be using more in this presentation, is the F sub zero value. And the F sub zero value is the equivalent amount of time in minutes at 121 degrees Celsius that has been delivered to a product by the sterilization process. So it's quite simple and intentionally oversimplified on this slide. But if I were to tell you I was going to sterilize your product at 121 degrees Celsius and I was going to run it for 15 minutes, you could assume that the F sub zero value during that 15 minutes was an F sub zero of 15. It's a one-to-one relationship as long as the temperature is at 121 degrees Celsius. However, if you drop that temperature by 10 degrees, you'll see that there is a reduction in the F sub zero. So of course, as you lower the temperature, you're going to get less lethality, and you'll get, in that same amount of time at a lower temperature, only 1.5 minutes of F sub zero. And then of course, then if you raise it from 121 to 124 and you keep the time the same, you go from 15 minutes to 29 minutes. So really F sub zero can be defined as lethality, or as it's a time, but you can kind of consider it as a lethality. And this can be done because of how well we understand steam and how documented the mathematics and the principles behind steam sterilization are. And this is taken from these formulas here, which I won't go over in detail, but you kind of see how you take the temperature and the z-value relationship, and you do some basic math, and you can kind of estimate at different time and temperature relationships what that F sub zero value is, what type of lethality can be expected from a steam sterilization cycle. So now that we have a little bit of a better understanding of terminal sterilization, the regulatory expectation, the resistance of how the D value and the F sub zero work for particular microorganisms, I want to shift a little bit and talk more about some misconceptions out there, specifically about evaluating terminal sterilization as an option for your drug product. So the main misconceptions are here, and they are that steam sterilization occurs at 121 degrees Celsius or higher. Cycles are typically 15 to 30 minutes. We need to use an overkill mentality, and highly resistant biological indicators, like the ones here, must be used. And just to visualize that a little better, typically when you talk about overkill and when people think of steam sterilization, when you think of biological indicators, everyone will say you're talking about Geobacillus stereothermophilus. You're talking about populations that exceed 2 times 10 to the 6, often, definitely exceed 1 times 10 to the 6, so 1 million. And the resistance, the D value that we talked about, is greater than 2, sometimes even 2.5. So if you go into the mindset with an aseptically manufactured drug product that I'm going to terminally sterilize it, and you're selecting these BIs, and the goal of the terminal sterilization process is to kill these BIs, you'll quickly find that to kill these BIs requires an extensive amount of time and extensive amount of heat. And we won't get into the mathematics. I'd be happy to offline with somebody if anyone had any questions about it, but we won't in this presentation get into the mathematics about how long it would take. But it takes a significant amount of time to destroy a biological indicator, or 10 or 15 or 20, depending on the load size, right, to show that these biological indicators were killed. And it's not really important to do that when you're focusing on a drug product, which I'll talk, a GMP drug product, which I'll talk more about later. And if we just keep this D value of 2 minutes in mind, and if you refer back to my D value slide, this means that every 2 minutes of processing at 121, so every 2 minutes of exposure at 121 degrees Celsius, this organism will be reduced by 1 log. Okay? So it's quite a significant D value number. It's a highly resistant organism, is what that's meant to mean. But if you look at D values of common organisms, and there's a lot of data from organisms from the food industry for canning reasons and things like that, specifically you'll see here on the left, E. coli, Salmonella, Staph, and Listeria. These D values, as you extrapolate them from their published values, which is their D60, as you extrapolate them and apply a D value for 80, 100, and 121, you'll see that instead of having D values of 2, you're substantially less than that. And what it means is really you would expect these organisms, even to achieve SALs that the industry expects, sterility assurance levels that the industry expects, instead of needing 12, 15, 24 minutes to eliminate the organism that's highly resistant, since these are so easily killed by the process, they're really destroyed on contact. And you really achieve high levels of sterility assurance just on contact. when you're talking about temperatures even at 80 degrees Celsius and 100 degrees Celsius. And you're getting in F sub zeros that are difficult to comprehend. Instead of F sub zeros and SALs of 10 to the minus 6, you're easily achieving F sub zeros of even 10 to the minus 500,000. So you get extreme levels of sterility assurance when you focus on practical D values as opposed to BIs with highly resistant organisms. So this is all within the theme of changing the narrative, right? So we talked about the misconceptions that are used, and want to shift now to kind of changing the narrative away from those misconceptions to what their thinking should really be. So the thinking should really be, when you have an aseptically filled or a sterile filtered drug product, and you want to evaluate terminal sterilization as an option, you should get away from thinking that you need those overkill mentality. So high temperatures, long cycle times, highly resistant BIs with high populations, and overkill mentality are not a requirement to terminally sterilize a drug product. There's essentially no bioburden for GMP aseptically filled drug products. And for that reason, the focus needs to be on killing bioburden at your facility, or typical personnel bioburden that could realistically contaminate a drug product, and to focus on killing that as a fail-safe to the aseptic processing. And there should not be this quick thinking to disqualify terminal sterilization as an option simply on the basis of product packaging, or of it potentially not being compatible with a high temperature at a high time, unless there's no choice or there's no other way to package it. And then, of course, unless there's data to the contrary that just completely eliminate any sort of heat treatment as an option. But the challenge is to kind of not be so quick for the industry to disqualify terminal sterilization without any sort of evaluation. And this changing of the narrative is not new, and is not something that, just Prince or just Altasciences is discussing, but it's out there. It's just not been overly adopted at this time. So again, in USP 1229.2, which I talked about earlier, they have a really nice section on bioburden/biological indicator method. And this graph on the right here does a good job of kind of explaining the 10 to the 6, the 1 million population of a biological indicator, and the long time it would take, presumably, right, at the steam sterilization operating temperature, whatever that may be, we'll call it 121 degrees Celsius, to get down to a 10 to the minus 6, or to get down to an industry accepted sterilization assurance level. The amount of time required, 15, 18, 21, 30 minutes, is a lot more, substantially more heat applied to that drug product than what would actually be required if you were just evaluating the regular bioburden of what was on that product, or even an extremely conservative bioburden approach where you apply a factor of 10 onto what the actual native bioburden is, and you use a value of 10 to the 2 or 100 contaminating organisms. And you can quickly see that just in minutes, at a temperature of 121 degrees Celsius, you've eliminated the bioburden organism down to sterilization assurances levels well beyond what the industry would consider acceptable. This type of bioburden processing is also discussed in ISO 17665, defined as the bioburden-based method, and it's further talked about in PDA Technical Report number 1 as the product-specific design approach. So, of course, when you're evaluating terminal sterilization as an option, you're going to ask the question: is the drug product compatible? Is the packaging compatible? Is there anything we can do to make the packaging compatible? But where some people get kind of lost in that is, how do you answer that? Is the drug product compatible, right? And I think what it appears to be is the thinking of, is the drug product compatible with 121 degrees Celsius? Is it compatible with 15 minutes at 121 degrees Celsius? Is it compatible to a cycle that will kill a highly resistant spore? So instead, we'd like to kind of bring to the attention of the audience decision trees. And there's 2 decision trees that we'll show. I'll quickly kind of talk about the one in PDA. And this kind of gives an idea about, if you have a heat labile material, is it heat labile, right? If it's yes, go ahead and use the bioburden approach, right? So that's kind of what this is saying. Don't focus on overkill, use a bioburden design approach, right? And then after that, it's not very clear about the next step. So it's quite vague, and it leads to some confusion by the user. Of course, if it's not heat labile, use an overkill approach. But again, it's very vague. A better decision tree, in our opinion, published by Jim Agaloco, I'll just spend a few minutes to kind of talk about this is the decision tree that we'll send anyone that's interested in a terminal sterilization of an aseptically filled product. And this decision tree really is less ambiguous and paints a clear picture that really focuses on key concept that performing any level of terminal sterilization is better than not performing any at all. So essentially it starts with, can the product be sterilized by moist heat using what Europe would consider a standard cycle, 120 degrees Celsius for 15 minutes. We'll call that overkill. If your product is able to withstand this, then of course everyone would agree to go with that option. If the answer is no, you work with Prince, you work with company, you work with your autoclave, and you work with us to look to sterilize by moist heat to minimum SAL of 10 to the minus 6. So again, this is thinking of, okay, what's the bioburden? What are we trying to kill? What's the population? And can we develop a good cycle, using this as a baseline to get to 10 to the minus 6? If the answer is no to that, you start talking about F sub zeros. So can we get an F sub zero of anything greater than 2? If the answer is that, then yes. If the answer is no, then you start kind of going through all of these different flow diagrams. But essentially, it even goes down to 100 degrees Celsius and 80 degrees Celsius. Whereas even if your product is compatible, it's not compatible at 100, it's not compatible at 121, but it's compatible at 80 degrees Celsius. The recommendation here is to even process at 80 degrees Celsius, because we know the D values of common organisms, and we know 80 degrees Celsius has lethality and will destroy organisms. So of course, any lethality that can be applied through a terminal sterilization process post aseptic filling is better than nothing at all. So this really challenges the industry to do a better job of evaluating terminal sterilization, and really provides a more clear overview of what types of terminal sterilization options there are, what temperature options there are to evaluate before so quickly eliminating or disqualifying a product for terminal sterilization. And then after all of this evaluation, if there's no level of heat that can provide lethality or sterilize the product, then of course, then resort to sterile filtration, aseptically process, and filling as the only manufacturing method. So of course, some things to consider when you're doing terminal sterilization of a drug product: potency and degradation, chemistry, what's the pH of the product post-sterilization, container closure integrity, is there any stopper movement, particulates, color changes, what label will go on it, expiration, storage, things like that. In companies, like Prince Sterilization Services, we help evaluate all these things to make sure that the process is not doing anything harmful or negative to the drug product. That's just as important, if not more, post-aseptic processing, if not more than sterilizing it. So we want to sterilize it, but we don't want to sterilize it if it's going to impact the actual use or ability to use the drug product or have an unwanted effect. So here I'll quickly discuss a case study, and it's more of just kind of a request that would come in about the terminal sterilization of a drug product. So I'll just kind of walk through how this process will work. And typically the first thing that Prince would ask for are safety data information. We would do a literature search, or we would ask if there's any historical information or literature for similar products, specifically, how that product relates to different temperatures and heat. We'd evaluate the packaging, and then we'd schedule for some development and engineering type cycles. There's 3 cycles we would typically evaluate, depending on the information that we knew about the drug products. If we knew that it was a heat labile material, we'd maybe skip some of the higher ones and go straight to a low temperature option. But if we really were going in blind, these are the 3 cycles we'd kind of evaluate. We would evaluate the standard overkill cycle, if you will, first, 121 degrees Celsius for 15 minutes. We'd evaluate a lower, same temperature, the preferred temperature of 121 degrees Celsius for 8 minutes. Then we'd also look at a lower temp option for a longer period of time. And what we would do is we would process the drug products, and we would do some testing at Prince, and we'd send back to the customer, and we'd basically be looking for, what was the effect of the sterilization process at the different sterilization parameters. What was the effect of that processing on the drug product and of the packaging. So in this, example for this case study, we can see that for this drug product at 121 degrees Celsius for 15 minutes, the container was okay, the packaging was okay, there was nothing in the vial, no particles or anything like that. And, all the miscellaneous tests, sterility, whatever else, endotoxin, all passed. However, there was a chemistry issue and a potency issue. At the 121 degrees Celsius, when we decreased the time from 15 minutes to 8 minutes, The chemistry was good, everything else was good, but the potency was still bad. However, the 115 cycle at 30 minutes, everything was good. So this would be an interesting one. And for this, it indicates that at 121 degrees Celsius, there's a potential potency issue at 121. So the drug product doesn't like that temperature of 121 degrees Celsius. However, when we lower it to 115, the drug product responds a lot better. So for this type of product, certainly you don't like to see potency issues at all. But this is an extreme case. We would really look at evaluating 115 degrees Celsius or below, just because we want to add a buffer. We don't want to have any risk of processing this drug product and getting too close to a known temperature that could cause a potency issue. So we'd really recommend this time to do a little bit more development, maybe even going down from 30 minutes to 15, going from 115 to 110, or anything less, and then increasing the amount of testing done for potency and chemistry to make sure that there's no harmful effects of the process on the drug product. So for this example, where it's a different drug product now, and we have done the same cycles, 121C for 30 and 121C for 15. Here, though, instead of there being an absolute large, defect or nonconformance at a specification for the potency, instead we have, it's right at the threshold, right at the borderline. So this would more imply to us that it's more of a time issue. As you the temperature could be okay, but the time is really harmful at that temperature. So for this type of product, if you really wanted to stick at 121 degrees Celsius, we were able to see that as you decrease the time, you eventually get to your desired container closure, chemistry, potency, packaging, particles, and miscellaneous products all passing at the temperature that we wanted just by lowering the time. So again, similar to the first one, you don't want to mess around too much with operating within the threshold of a drug product's potency. So we would increase development, do another 3 or 4 or 5 development cycles, and we would maybe go to 120, 115, 110, and we'd evaluate different time temperatures just to feel good that nothing was happening to the potency at those temperatures. It's always a balance of sterility assurance and of course, there's cost considerations to it. So just for that last example, some things that you are also looking at with respect to sterility assurance specifically are F sub-zeros and BIs. And for all of these examples, you can see that after the full cycle would be done, you would get F sub-zeros even at 120 degrees Celsius of 8 minutes. We would be able to map what the F sub-zero is, what that lethality truly was at the end of the cycle, and whether or not biological indicators were killed. So for this product that we did these 3 cycles on, all 3 cycles had adequate F sub-zeros. All 3 cycles had biological indicators that were all killed after the process was done. However, only one of them met the other important aspect of it, and that's that it did not impact this drug product. And that was by reducing the time. So there are options to reduce the time, there are options to reduce the temperature, and still get the desirable effect of sterilization. So after you did those studies assurance, which I just presented, that would really conclude what we would call the development or the engineering phase. And once both parties felt comfortable that the drug product and the packaging system was compatible with a given time and temperature sterilization process, we would move on to what's called validation. And validation is kind of summarized here. There will be a protocol signed off on. The sponsor will send the material that will be used for validation. There will be the maximum and the minimum load. And then there's a full temperature map and biological indicator placements. So at this point, you're really putting things called thermocouples, and you're mapping the temperature of the load items, the liquid, for the actual F sub-zero value, for the actual sterilization effect, the heat penetration that's getting to that product. And you're also placing the biological indicators and making sure that you are getting biological indicators that are being killed through the process. Once that's all done and the data is reviewed, a final report would be generated. It would always be recommended that all of that testing that was done during the development and engineering cycles would be repeated to a certain scale. So again, you would confirm that the potency, the chemistry, the container closure integrity system, everything was performing as expected. And at that point in time, we would accept the validation and it could be used in a submission. Post-validation and submission, we move on to routine and commercialization of the drug product. So this assumes the validation report's complete, cycle recipes are saved and locked, batch records are saved and locked, and then we're ready to produce routine commercial loads for this drug product. So just a real quick summary of the benefits of terminal sterilization. Overkill is not required. This will enable more products to be compatible. It's highly recommended not to focus on 121 degrees Celsius for a defined period of time. Evaluate different temperatures, evaluate different times is highly recommended. Use that decision tree as a guide. With terminal sterilization, the process is lethal. So with aseptic fill and sterile filtration, you're really just physically removing, hopefully physically removing bioburden from the product. With a terminal step of any degree, 80 degrees, 100 degrees, 121 degrees, you're actually providing a lethal effect to the organism. And that's defined, and you can define that sterility assurance level. Of course, if you do this from a clean room management and aseptic handling and aseptic processing standpoint, you're less dependent on your controls. So you're less vulnerable to process simulation and media fill type things if you're performing this downstream terminal step. Much easier conversations with regulatory authorities, much easier conversations during inspections of your filling equipment, your filtration validations, your clean rooms, your sterilization program of your stop ring and crimping and inline manufacturing equipment. Just much easier conversations and peace of mind, if you will, with controls if you have a terminal step. Of course, much lower risk for an adverse event with respect to sterility, which is safer for the patient, which is what this is all about. We'll reiterate here that any F sub 0 is better than none at all. And that's just a technical way of saying any exposure to a terminal sterilization process is better than not doing it at all, provided there's no harmful effect to the drug product, of course. And again, just a reminder to not let perfect be the enemy of good, right? Just don't focus on overkill. Focus on, is there a heat process? Is there a terminal process that we can apply to make this a safer experience for a potential patient? So of course, the question, can we do better? And if patient safety is the number one goal, then it's clear that terminal sterilization needs to be more widely adopted.
Thanks, Derek, for the great presentation. Now we're going to move over to our Q&A session, and we will be taking questions from the audience.
Thank you, Andrew. I see there are a few questions for you. The first being, why are potent compounds manufactured aseptically?
Well, that's a great question. The main concept of clean rooms is to limit exposure. Exposure works like a two-way street. While we want to limit particulate exposure to the product, we also want to prevent exposure from a product to the operator. Potent compounds are defined as having high pharmacological activity at low concentrations. And during manufacturing, these compounds can aerosolize or vaporize to become airborne particulates. So we want to use engineering controls and good practices are utilized by clean rooms to limit exposure of these potent compounds to the operators.
Thanks, Andrew. And how can Altasciences check for microbial contamination during manufacturing?
While we cannot guarantee sterility during manufacturing, we can take precautions during during, and post-manufacturing. Prior to manufacturing, we regularly clean and test the rooms and all equivalent equipment in between manufacturing campaigns. High-quality GMP-grade material is also sourced for our campaigns. During manufacturing, we use sterile spatulas, pipettes, and containers to weigh, dispense, compound, and fill and package into its final container. After the product has been packaged, a bioburden sample is pulled and tested for microbial contamination. After packaging, the product is then shipped out for terminal sterilization.
Thank you, Andrew, and thank you, everybody, for the good questions.
All right. Seems I have a few questions for Derek as well. Is product sterility testing required after terminal sterilization?
Yeah, so with respect to product sterility testing, if the sterilization cycle is validated and bioburden of the product is controlled and understood, then sterility testing is not required and a process called parametric release may be utilized. Therefore, any sort of USP or EU sterility test is not necessary. And this is a popular option for validated processes as it saves customers significant time and money when releasing their product from a terminally sterilized cycle.
Perfect. Thanks, Derek. Another question here. Can you talk more about how validating a TS process works?
Sure. So validations typically consist of 6 cycles, 3 being a minimum load of the product and 3 being a maximum load for the product. Bundled into there, there's also cycles to verify the performance of the sterilizer or the autoclave, which include leak tests, Bowie-Dick tests, empty chambers, and other types of development and engineering cycles that would be required. So a validation can typically be anywhere from 7 to 8 to even 10 or so sterilization cycles. As part of that process, things like thermocouples to measure temperature and BIs to measure microbial lethality are used and put into the product to show how effective the terminal sterilization process is.
Okay, great. Well, looks like we have one more question here. Why is steam the preferred modality?
So steam sterilization is the preferred modality and always referenced as such in the regulatory authorities, primarily because it is environmentally friendly, non-toxic. There are no harmful residues. It's broadly accessible, and the lethality of steam is well understood and established. So as such, efforts should be made to use steam as a sterilization option whenever possible and to not be dissuaded by long cycle times and high temperatures.
Okay, great. So I think that's all the time we have for questions. We will personally answer any of the remaining ones after the webinar. If you have any other questions, you can contact us using the email address on the slide. Thank you for listening and your participation. And Derek, I would like to thank you again for your expertise on this matter.
Sure. Thank you, Andrew. Thank you, everybody, for listening.